Today is Thursday, March 13, 2014. My name is Martin Frank, and I'm here in Chicago at the University of Illinois College of Medicine in the Department of Physiology and Biophysics to interview a friend and colleague, Brenda Russell, who has been a member of the American Physiological Society since 1991 and a professor here at the university since 1988. Brenda's research has focused on cardiac muscle remodeling and regeneration. In the course of her career, she has published over 100 manuscripts and papers. Brenda, it is my pleasure to have this opportunity to talk about your career and learn from you, and the opportunity to have it shared with our readership and our listenership. So welcome to the Living History program, and I look forward to hearing about your career.
I'm happy to be here, and although I did have second thoughts sometime yesterday, you were already in town, and I'm looking forward to the conversation too.
Sounds good. Thank you very much. Why don't we start by talking about your early years growing up. What was it like growing up in World War II England, and how did you get interested in science during those early years?
Well, of course I don't remember World War II, but it had a huge impact on my parents' generation. My father was a pilot in the RAF. He was actually a Battle of Britain pilot, one of Churchill's few, and of course he lived through the entire war, not just that early episode. Mom was a farmer's daughter and wonderful mother, and the at-home person who held it all together. Early years in science, I think you asked. So I brought something that I don't know why you keep the few things you do, but this is one I kept, and it's incredibly funny. My grade school, private girls elite, upper middle class, I think we used to call it, Science for Girls. And I'm going to read you what it says, and you will wonder why I ever became a scientist. The syllabus is designed to help girls to understand the scientific principles underlying modern ideas and appliances, which have done so much to relieve household work of its drudgery, and will give them a grasp of the main points of healthy living. That is science for girls. And they have lovely things. I'll show you this. A vacuum cleaner. Science for girls. My parents' idea of science for girls was how to be a better mother and housewife. And they certainly didn't. They weren't scientists, they didn't go to college, they were typical of their generation, you didn't need to go. How I became a scientist is a little bit of a mystery.
Clearly, as you told me, most girls were not encouraged to go into science, they were encouraged to get married. But clearly something triggered an interest on your part. You went to college, you went to University College London, and you wrote in what you had provided me an indication that you decided to get into physiology. And why was that?
Well, I loved numbers, and my earliest memories are of wondering about these numbers and what they were all about. And I loved understanding things and asking questions. And Daddy was a very independent thinker and numerate. I was a naughty little girl in this private girls' school. And the headmistress...
Could we talk about that instead?
No.
Oh.
No, we can't. I mean, I let off stink bombs, and I was really naughty because I asked teachers questions that they couldn't answer. And then I would go and, I don't know, come back with an answer and tell them I thought they were wrong, and of course they were wrong. And the headmistress got tired of having me in the office and getting misconduct marks, and equivalent to 100% in straight A. And so she told my father that their little girls' school was not an appropriate place for me, and he should send me to one of the private boarding schools, or I grew up near Cambridge, to one of the private day schools in Cambridge. And Daddy said, that's a waste of money. She's a girl. And so instead I went to the local grammar school. And that was coeducational, and the science teachers were male and extremely good. And I just loved what I was learning. So as the teachers from this school, and you have to understand the class mismatch. That's like it was a wonderful school. Academically, it was marvelous, but the children there were the farm laborers' children.
Okay.
And my family, as it were, owned the farm. And so it was culturally very revealing to me to be in another social group, and it broke up some of my concepts. It was really hard at the time, living in two worlds and speaking two dialects, local and the other one,
right.
BBC. And it was really because of the teachers I kept learning. And I actually left school and didn't go right to university. I spent 6 months in California staying with my father's cousin. And it was being in California and seeing that all women were not just moms.
Okay.
And seeing, as it were, the new world and opportunity. And everyone there said, oh, you must go to university. I'd applied and been accepted, but I hadn't gone.
Right.
And my father thought I'd go see the world and he'd pay for that, and I'd come back and get married to the eligible bachelor that they had picked out for me. And it didn't work out that way. I went to university and married an American, and subsequently came back to the States. So,
how did you? One of the things that I thought was interesting is, for a physiologist like myself who read Katz and Milady and read Huxley and Hodgkin and things like that, is you went to University College London, you got your bachelor's, BS in physiology, and part of the honors program was reproducing the works of the famous physiologists that we all grew up knowing, and here they were right on your doorstep.
Yeah, and why physiology, you asked as well. My favorite subject in school was actually physics, and the second thing I wanted to do was to be a doctor. But women in England, in my vintage, this would be, vintage is a good word.
Yes,
yes.
Like a fine wine.
Exactly. Starting university in 1962, women in medical school were probably 2% of the class. In engineering, there were none. Physics, it wasn't, I mean, you couldn't go. They didn't, oh, you're a woman. It didn't matter that you'd got good grades. You weren't going to be accepted. And I went through the curriculum and all of the courses, literally from anthropology to zoology and back again, and found physiology describes itself as the science of life. And I thought, oh yes, I want to do that. And that's why I applied to physiology. But you asked about the program. Another thing that goodness knows why I kept this, but I did. Put those on the floor for a minute. This is the folder that we had to produce. There were two like this for the third year, final year of the honors program. And it's all falling to pieces. And of course, it's all written in ink. I don't know how to show this handwritten in ink. And it has on the front page, it has my candidate number. And this is part 1, nerve, taught by Katz, Niedergerke, and Milady. And
they taught the course?
Yes.
Awesome.
And then the second part is muscle. And, you know, I flagged where the Huxley thing is. So what we did, I don't know if you can see this. Oh, I love this. It's scribbled out. I still do that. So this is the cats. Spontaneous subthreshold activity at the motor end plate. And this is 29th of October, 1964. And we did these recordings, you know, on sheets of paper, all handwritten, histograms built as you went with little hash marks, columns, quite neat for me, I'm not terribly tidy, hand-plotted curves and so on. This is what we did, end plate potentials, for which, of course, Katz got a Nobel Prize. Here we are using his instruments and recording miniature end plate potentials as undergraduates. Undergrads.
Yeah.
Quite remarkable. So, and I like the end of that. Hand-plotted Poisson distribution, curve fit, and it didn't fit because I got the theory wrong. So I have a note here. Note, the theoretical curve opposite is not correctly calculated. The peak heights arrowed, theoretical curve, this curve is wrong. They taught us how to be honest and say what you'd done wrong. Should be normally distributed about the peak as a mean, and so forth. And I remember that because I got it wrong, and mine were all down. My theory was up there, my experimental were down here. Actually, my experimental were really good. My theory was not. And they liked it, that I was honest enough to give the real data and say it didn't fit the theory. And then they go, oh, you made this mistake, and you did fit it. And they told me afterwards that trust in my honesty in data reporting was something that they wanted students to learn. And then this is Huxley, which is the other kind of fun one looking back. So this one is sarcomere spacing from his home-built interference microscope. And that experiment didn't work very well.
Which is the important thing to recognize. And a lot of students don't, that experiments don't work all the time.
So it goes on with pages and pages and force velocity curves and so forth. So that's what it was like. And I'm actually impressed, not with my student work, but that Nobel Prize winners took the time to work with this group of undergrads and teach them how to do the experiments, how to analyze the experiments, how to deal with things not coming out the way you thought they ought to. And we knew how they ought to.
Right.
So that was a very fine education.
Well, I think it's a wonderful stimulus to encourage people to pursue science, to have people like that in the lab with you, helping to teach you. And I think, I'm not sure we all our esteemed faculty put as much time into educating undergrads, let alone graduate students, as they should today.
But they were role models.
And
sure.
I've had the pleasure of meeting several of them, and they still are role models today. As far as your own academics, you finished with an honors in physiology, and you didn't go on for your doctorate, and you did briefly say that you came back to the United States because you fulfilled your parents' dream. You found somebody to marry, unfortunately, not the person they had selected, but Bob Eisenberg, Robert Eisenberg, who many of us know as well. And you followed him along and didn't pursue your career for a while, but tell us a bit about the travels of being a trailing spouse.
Well, I'm a positive person. My priority at age 21 or whatever it was definitely to be, being married and having children was really important to me. It was important to my parents, but it was important to me as well. And I would make that decision again. I don't think you have to choose between the both. But when I did have to choose, I chose being a traditional mother and housewife and trailing spouse. Happily.
So that book helped.
That book didn't.
The Science for Girls.
Oh,
helped.
the Science for Girls helped. Yes,
Right.
that's
Okay.
right. Science for Girls was good. So I knew
How to cook.
I knew how to vacuum.
Okay.
So that was important, and it was very exciting. I worked as a technician in a lab, and kept doing independent work. And that early stuff, when I was supposedly a technician, was published in Science and Journal of Cell Biology. I mean, and I was doing it part-time with the enjoyment of the faculty around, who didn't treat me like a technician,
Mm-hmm.
probably because I didn't behave like one, and probably because I didn't think like one. And so many people kept saying, you should get a PhD. You should finish your PhD. If I'd stayed in England, I would have been a graduate student with Huxley. And I left that. So at some point, actually it was after baby Ben was maybe a year old, and I found out that there was a lot of empty time in the day when a baby's sleeping, and
Mm-hmm.
and vacuuming only fills up so much of it. And I wrote back, and the other funny thing, I was turned down as a graduate student at Duke and at UCLA because I didn't have the prerequisite undergraduate course credit hours. I mean, this
Right.
didn't count. So I would have to go back and do all kinds of things, nerve muscle physiology, and I'd had nerve muscle
cats
from
and-
Huxley and cats, and it seemed pretty silly to think anybody could do it better than they had. Actually, Ernie Wright, another English physiologist at UCLA, said, why don't you reapply as an external student, because there are no courses in the British system. You just have to do research, which you've been doing and publishing. And I wrote back, and Huxley volunteered to be my advisor, which freaked me out. And
by then, he'd already won his Nobel Prize.
oh, he won it while we
While
were
you were
there.
there.
Yeah, 65, 60, something like that.
Yeah.
Yeah. So, so that meant writing the thesis and finishing the research and submitting it. And Huxley was extraordinarily demanding, and I think he had either no other or one or two other PhDs in his career because he was impossible. His standards were too high. But he would enjoy what you're doing with living history. Huxley is a real fan of the history of science and the history of physiology because of his grandfather, Thomas Henry Huxley.
Right.
And he loved archival material. So for my thesis, he kept saying, what are the earlier references? And I joke that the only reason I got my PhD, one is I had a baby and I said, no more experiments, I'm home. And it's hard to answer that. And the other one was I read the German anatomy of muscle in German and translated it and got some references on sarcomere spacing that he didn't know. And then I went back and I got one from 1712, Van Leeuwenhoek. The very first time the new lens was used, and it's a really funny paper, and it showed the striations. How much time do we have? I'll tell you how funny this paper is.
For a drink later tonight.
Okay. Because Van Leeuwenhoek was trying to look at the heads of sperm from a sperm whale and look at the little critter in the head that
Right.
the male planted that seed, and they didn't get any sperms, but they got the muscles
Okay.
in the correct anatomical part of the sperm whale.
Sounds good.
Yeah, so that's a funny paper. But I got that reference, and we'd have all these conversations about history of science, and that he could talk about.
Okay.
It was not easy to chat with Huxley.
Well, I have some stories, and we can talk about them later with him. After you got your doctorate, you were looking to get support, and you applied for an American Heart Association and a Muscular Dystrophy Association, and you ran into the nepotism issues at UCLA. Talk a little bit about that, and talk about the experiences of having dealt with that era of science.
So, I got both those fellowships, and sponsors to do the work. I had a PhD from a Nobel Prize-winning physiologist. Bob Eisenberg was in the physiology department, and the University of California, well, probably all state thing, nepotism meant a husband and wife could not be in the same department. And since he was in that department, I could not be in physiology. Credentials didn't matter. And Sherm Melenkoff was the dean at the time, and he wanted to use our case as one to take to the state to appeal. They're saying you should be able to be employed on your own merits. And in the end, they didn't, because they had another couple who both had an appointment and then married, and they tried to throw the woman out. And at the time, it didn't seem strange to me. That's odd. It's like girls should be in the kitchen, and you were lucky to be doing science at all, kind of be quiet. But the optimism in me, I mean, how these bad things turn out well. Now, I knew Sherm Melenkoff, so the real obstacle for women in science wasn't that silly rule, because you could go and be in another department, but it was childcare and managing hours and leaves and the practical. I'm very practical. So I was actually the president of the first UCLA childcare center, and Sherm Melenkoff made that happen. Okay. I went to him and he goes, well, you know, I know you, and I think you can be a postdoc and run this brand new operation. And Ben was in the child care, and I went and spent like 2 or 3 hours there every morning, and then went to the lab. Yeah, I've always done the juggling.
Juggle them.
Yeah, but that was the rate limiting. It wasn't the legal appointment. The rate limiting was quality child care. So you make things happen that needs to happen.
You were at UCLA for a while, and then Bob got a job as chair of physiology at Rush University Medical School. And
right.
You came east from LA to Chicago. Tell us a little bit about that transition in your career and setting up your independent program.
So that's another trailing spouse. Actually, at that point in time, I seriously considered leaving science. He had a substantial appointment. I didn't need to work. We by then had 3 children, and I wanted one more, and we eventually had a 4th. And the relocation and schools and a new house and getting to know a new neighborhood. Don't underestimate the work of the traditional second person in the family. It takes time and effort, and you need to be there. And so,
especially with little kids.
With little kids. And what I wanted was part-time employment. And at that time, it was only full-time, and full-time was, I don't know, a 60-hour week. And I said, it's not workable. I don't want to do it. And again, I couldn't be in physiology because you can't report to your spouse. So I was in pathology. And I arranged for something like that. It was a fewer hours, and I was set up a clinical diagnostic lab, was then on special stains for diagnoses in muscular dystrophy and some neuromuscular disorders. And I had research funding, maybe MDA, muscular dystrophy. And I was really keeping my hand in the lab, but parenting was, running the home was the major If I think of those times, I think of home and not lab.
Mm-hmm.
And I remember signing the contract, and I wouldn't sign the 60-hour contract, and that's the only one they had. So I just scratched it out and wrote 30. I don't know what they did with it.
They probably didn't read it, like most contracts, until they needed to bring suit against you.
Yeah, so
but they didn't.
So I have another funny one at Rush, these women's things. So I'm pregnant with Sally, and by then I'm tenured, and I want a leave of absence. I want at least 6 months maternity leave. And I was then in pathology, and the chairman brought me in. He said, "Do you want the good news or the bad news?" And I said, "Bad news." And he said, "You can't have a leave of absence. You will lose tenure, and you will have to be reappointed, which will be very difficult." I said, "Well, what's the good news?" He said, "Well, the good news is that's really not in compliance with law, so we're going to have to pay you full time." And the lawyers are fixing that. Because I was the first tenured faculty to be pregnant. That's not that long ago, but, you know.
There were not many women in academia.
This is true.
And we still need more.
And I mean, I think it's funny.
Yeah.
I mean, it was funny at the time, and everybody thought it was funny. But I'd asked for the leave. You know, it took someone both to be tenured, be pregnant, and ask for the leave to make those changes.
So in a way, you were a trailblazer.
No, I was just practical. I was not going into work with that baby.
Okay. Tell us, and we haven't really talked much about the research that you were doing before you came to Rush and at Rush. Tell us a little bit about the research you did. You and I got to know each other because of the expertise you had in microscopy. But I know you did a lot of stuff.
I have a photograph to show you. I'll put these back on there. But I haven't shown you that.
You can put that in the video.
I can put them in the video. This is when you first met me.
Right.
Brenda with the electron microscope. Yes.
Because I remember bringing you in for a study section when I was running the physiology study section because of that expertise.
So what I was doing, I liked the electron microscope because it's electromagnetism, it's a physical instrument. And the instruments now, you just press a button and it all auto everything's. Then you had these, literally, I couldn't reach the top ones. I couldn't look at the bottom, and I wasn't, it was built by a Siemens, a tall German. But you had to twiddle these top knobs, and these are each magnetic lens down the column. And if you didn't understand what you were doing, you couldn't get the image in focus. So it wasn't just preparing the tissue and thinking of the experiment, it was actually acquiring high-quality images. So I learned how to do that at Duke and continued to do it at UCLA. And being quantitative, the other thing I did was not just take the photograph, but measure, figure out ways to measure all of the components. So you didn't just say, "Isn't that a pretty picture? Look at those blodgy round things in the corner. I wonder what they are." You found a mathematical analysis to turn the image into numbers and therefore quantify the components. And with that, I could go back to trying to understand how does muscle adapt. And so started looking at normal muscle, skeletal muscle, cardiac muscle, and analyze the various components, but thinking like a physiologist. What do the sarcomeres look like? How much of the tissue is sarcomeric? How much is mitochondrial? We knew, I mean, my PhD thesis was on the T system. How much is T system? What's the element of the calcium storage in the SR? And in different fibers of the body, the proportions vary depending on the function. Okay, that's nice. that's how do you turn that into a hypothesis? You drive the muscle in new functional patterns and see if the organelle ratios change. And I was at an international conference and became reacquainted with Stanley Salmons, who'd been a classmate in London. And he'd built a stimulator, he was an engineer by background, to pace the nerve and pace the muscle. So he'd got muscle that had been paced. He fixed the tissue in England, shipped it to California, and I analyzed the compartments and showed that if you exercise a muscle, it grows more mitochondria because it needs more oxidative capacity, for example.
Right.
And then looked at the speed of contraction, and that tracks with the SR and calcium handling. So that era was very collaborative. It was international, and it was what can I say, sort of, those pictures are now in Grey's Anatomy. That probably says what you need to say. It was the opportunity to understand what was happening, but then do it in a scientific manner. Mm-hmm. And it took a lot of people, and it worked. So that was that era.
Clearly your interest in physiology continued while you were in the pathology department at Rush, but in 1988 you decided to, I guess, officially become a physiologist by joining here, University of Illinois, the Chicago Medical School here, in the Department of Physiology and Biophysics with John Solero. And so how was the transition coming to all of 2 blocks, or 3, from Rush to set up lab here?
Yeah, well, you're being very polite. I mean, Bob Eisenberg and I got divorced, and by that time I was in his department because those rules had been changed. So it was now the department head was my ex-husband, which seemed a little uncomfortable. And I had grant funding, and I heard that John was going to accept the position here. And we had joint custody of the children, and both wanted to see them. They were then, grade school, one in high school. So it was an ideal solution, and of course I'd known John through scientific connections, and so I was very happy to come here and move my lab. And well, actually some of the stuff, it is just 2 blocks, and some of the heavy things were moved on a truck, but the precious things we did roll on a cart across. And so it was a very, the lab part of the move was easy. The life was not. Reestablishing, divorce is awful. The science was not the issue, it was all the home things.
Right.
And very disappointing and stressful, and worst time in my life, for sure.
But coming here, and John was very welcoming, and I think...
John's great. John is one of the best people everybody knows. So.
But I think one of the hallmarks, once you got here, was the collaborative, interdisciplinary aspects of your research, and how you reached out to multiple departments and multiple divisions. And I think it would be nice to talk about that, because I think it's an important thing that if we have any young people listening to this videotape, for them to think about in their own careers as well.
I mean, if you learn from these heroes of physiology, they did not think they could do everything themselves. They always talked to experts and listened to others. University of Illinois, Chicago is a big university and has an engineering college. And one of the collaborations I set up was with Tejal Desai in engineering, bioengineering. Just earlier this week, I was talking at a chemical engineering conference. And the last slide, I was looking at who my collaborators were and where my students have gone or come from. and is remarkable. So clinically, cardiology one would expect, but it's also obstetrics and internal medicine. In the professions, chemistry, I had a joint grant with a chemist, engineer, clinicians, multiple collaborations. It's just too complicated to know everything and do everything well. You can't. And then my students have come from, physics and chemical engineering and biology and biochemistry, and have gone to medical school, dental school, nursing school, physiology, but they've also gone into industry in all different walks of life as well. So you can't do it. It's a team, and you need to know what you know. You need to be even more conscious of the things where you're an amateur and someone knows more. And then the difficult part is to know who in that other field knows what you need to know, and then what kind of person they are, because not everybody can collaborate. That takes an attitude of mind rather than an expertise. You can't get it from what you read in a paper.
Right.
You have to meet them and see if you can exchange, and you have to know that. Now they'll know this, you have to know someone is going to say, I don't know that, I don't understand that, can you explain it to me? Then you know you've got a colleague. The people who goes, yes, I know it all. Mm-mm.
One of the collaborations you mentioned led to the founding of a small company and some patents. Tell us a little bit about that.
The patent process or the company?
The patent process is probably too dense to be talked about, but talk a little bit about the company and how it came about.
So the company's name is Cell Habitats, and this is with Tejal, and she also now has 3 children, so she's sort of a generation younger than me, but sort of watching it still work is really nice. We named it Cell Habitats because I'm growing cells in a culture dish and feeling like the cell's not very comfortable. Like you put a kid in an uncomfortable car seat and strap them in wrong, they're going to scream. Mm-hmm. And I'm sort of saying my cells aren't comfortable in this dish. Muscle cells really want to pull on the ends. They don't want to just lie flat. It's like taking an egg and cracking it into a Teflon pan and this thing spreads out. It's not like a muscle. I want to have something it can hold on to. And she's a microfabrication engineer. So she said, well, what size and what would it like what do you think it would like to hang on to? And I sort of draw it on the back of an envelope. She goes, that's easy to make. And she made it, and we plated the cells. And it's like the funny things you do. She made them and gave them to my lab, and we plated them. And half of them worked and half of them didn't. And then we realized some of them were upside down. So the sort of side with the projections was up, and the cells hung on to them and beat more. And some of them were on the back, like, oops. So we learned to code for that. And that was just the simple idea. So that became then how do cells attach in 3D culture? And we have one patent for that. And then the next one is you can put drugs or peptides or hormones in a cell-sized structure, and both the structure gives cues to the surrounding cells and the chemicals do as well. So these can be injected into damaged heart and regain function by adding stem cell homing factors, anti-apoptotic factors, something for the cell to hang on to and behave in a different manner, because the cells surrounding the structures are cued by local mechanical forces. So that's the second patent for that and the basis of the company.
All right. Well,
and if you've got $5 million, someone out there, we know what to do with it.
Would you like to make a pitch?
I just did.
Okay.
Cell Habitats. not CELL. Habitat, keep the hell.
All right.
Cells
One of the things, we talked about it a little bit, working with Huxley and Katz and Nita Gerke. But you also touch base with some of the other leaders in muscle physiology. Paul Horowitz, who I think you met at Duke, and he was at Rochester as chair of physiology.
Mm-hmm.
Williford Momartz
Mm-hmm.
at UCLA. I think you worked with Glenn Langer.
I did.
Who I know, my research, I used to read his papers all the time for calcium fluxes. But if you want to talk a little bit about some of the people that you interacted with. And I know you also mentioned that when you and Bob got married, your upstairs neighbors were
The Armstrongs.
the Armstrongs.
Yeah, so I wasn't, this is the physiology department at Duke
Okay.
in 1968. And I'm not on the photograph because I
You
was the
weren't part of the
trailing, I
fam-
was the trailing spouse.
Were
But-
there any women in there?
Clara Francini Armstrong
Okay.
is the only woman. Is she? I think she's the only one. And how many? I don't know, 20 men. Dan Tosterson was the head of department,
Right.
famous.
Former APS president.
Yes, and on to Harvard and
Right.
so forth. Paul Horowitz was one of the people who treated me like a scientist and not just a wife. And I had great respect for him. He was one of the ones who encouraged me, said, this is quality work. You should continue your career somehow. So he was one of those people. And put that one down. And then here's a more fun picture of the Duke bunch. This has Paul Horowitz with the bow tie, and Bob Eisenberg and myself, and then Clay and Clara Armstrong, and the tall fellow is Peter Gage. And that's happy Duke days. And the Armstrongs were both, did postdoctoral fellowships with Huxley and overlapped. They actually lived above us, well, because there was a connection and it wasn't random, they lived above
Right.
us. Clara arrived pregnant, and she had to do 2 years out of the country to convert her visitor's visa into a visa. That's how it used to be.
Right.
Even though she was married to an American, she had to leave and reapply. So we knew them there, and then we went to Duke, and the year after, they came to Duke. And so, and last time I saw Clay and Clara was, I guess it was the biophysics meeting in Philly last year.
Okay.
So they've been lifelong friends. Woods Hole was another place. So that was science. And of course, Clara was doing muscle electron microscopy. So
Right.
we never published together. We were both at Duke and interacted in the lab. But she was on maternity leave then, which is probably a good role model for me
Right.
to see that it could be done. But they were really good friends and good scientists and nice people to know. Momartz at UCLA, I remember more that he had his wine importing company and sold wine to all the faculty, which these days I think would have been considered something you don't do.
Probably not.
I have another snapshot. These are from my old crinkly albums, which is why they've got torn pages on the edge. I don't know how these are going to be cropped. But the various people, I have to turn it this way around to see, Dick Chan and Clay and Richard Adrian. Richard Adrian was the external examiner on my thesis committee from Cambridge. And then this other panel here is Hugo Gonzalez, Martin Morad, and Martin Schneider, all sort of famous physiologists in their own right. And these were friends.
Right.
And I mean, Hugo was in London with Huxley.
Okay.
And Martin Schneider was at Duke. And
Right.
of course, Morad was UCLA, and Morad is Morad.
Yes, he is.
Shall we talk about other kinds of people in science and issues that women had to deal with, or shall we?
I think we do that off tape.
We do that off tape. I think I should say something, though. There weren't many women in science, and there were a lot of men. I saw that
Right.
Duke photograph. So it was difficult for the women and the men to know what the role model, who were you? I was there as a scientist. I wanted to talk science. But it was confusing for the men. You know, you were a woman, so you were there as a woman. And it took a while for some men to respect women. That's why people like Paul Horowitz and, Glenn Langer was another one, were exceptionally fine human beings and treated people in a way, the right way for the situation. So there were some real gems.
Right. What were some of the most interesting or most unexpected findings that you think you made in your career?
The next one.
The next one.
The next one.
Okay. We'll leave it at that. Tell us about scientific meetings and their role and how they differ from scientific meetings today.
Well, the first meetings, definitely at the first meetings, would be 2-3% women attendees. And the advantage of that was everybody knew my name. I mean, easy, you didn't look
Right.
like anybody else. The difficulty was you didn't have they knew you because you were a woman, and that wasn't why you were there. In the meetings themselves, they were lots of little dark rooms with slides, glass slides being projected, and 3x4s or 2x2s, and not much conversation or exchange a short question time. And the posters weren't introduced until, 1980s maybe?
Probably, yeah.
And now we think of a scientific meeting as lots of posters and lots of informal interactions, which are really good. So the actual format of science meetings is more collegial and more interactive, more opportunity for young people to meet their peers and the senior people and vice versa. So I think meetings have become more friendly and more informative.
Okay. While you have been involved in the APS, more on the publication side than any, I know you've interacted with other societies as well, and I think it's worthwhile to talk a little bit about the role of societies in your career as well as career development of other young people. And by that I'm including you as one of the young people.
Thank you. Well, I didn't travel much, and many of the society required additional travel. I would go to maybe one scientific conference a year because childcare,
Right.
or, it meant leaving the children with grandparents and then going to a meeting. Grandparents weren't in town, or leaving them with Bob, and then
Right.
go. It wasn't, I didn't travel much. I think that's why I didn't get involved in professional societies, because that was an extra travel that wasn't the right thing to be doing. Later on, though, I have done it. I think they're very important. I was really active in the American AAMC, American Association of Medical Colleges, and was on the founding board and then chair of the GREAT Committee, Graduate Research Education and Training. That organization, all of the FASEB, they speak for they speak for everybody else, so that the politicians and the newspapers have some place to go to get a credible response. And 1,000 people writing just gets in a check mark on a politician's box. But a president of an organization can go and be interviewed and have a present, really makes a difference. And the people in the professional organizations know about the subject and they care and they're articulate. So it isn't easy to be a position like yours, but it makes a difference that we have them. So I'm enthusiastic about professional organizations. And, you know, you need someone to go be there, do understand the issue and respond.
Well, I think it's critically important to have people to speak on behalf of the investigator community, and I think it's increasingly important for the investigator community not to hand it off to other people. I think we all have to get more involved, and hopefully coming generations will be doing that because of the situation we find ourselves in terms of research funding.
Yeah.
What advice would you give to a young person today who's thinking about pursuing a research career in a
For me, it's been the most fun, best thing to do. And I say if this is the passion, you enjoy this, you should do it. If I'm just taking on a new PhD student, and the reason he's joining the lab is he's been working in the lab for a bit in a master's program. He just loves the science. You can tell he's enthusiastic about this next thing that's going to be great. And you try a brand new experiment, you guess what might happen, and it works. And you know that you're doing it because there's an application. You're answering a question, the question's important. But it's fun, and it's still fun. And if you can get that enthusiasm, if someone senses that enthusiasm, this is what they should do. Get the PhD. Will they all continue in labs? No, they won't. But what they will learn is how to turn that fun in. I had a student actually this Monday defend a PhD, and she was saying, you know, what she'd learned in the time doing her PhD. And it was how to ask a question, what you do when you don't get the answer you expect, how you regroup, come back, ask another question, learn to organize, write up, explain, and just what you learn with a PhD. It's invaluable. The people who leave the lab and go, they're not all going to be professors. They're going to be successful in whatever walk of life, because this formal training of a doctor of philosophy, it isn't about the technique. It's about the independent thought and the analysis, judgment, and comprehension of a field and a way to explain it, communicate to others. You can't lose by doing that. It's a winner. But you have to have the passion for what you're working on.
Critically important. We're about to wrap it up. I'm sure we did not touch on everything that you expected me to ask you, but is there anything that you would like to add before we wrap up the
Well, I'm
interview?
looking at my pile, and I realize I do have one last picture.
Okay.
This is the balanced life. So this is Professor Huxley. He told me to call him Andrew. I never called him anything but Prof.
I call them Sir Andrew.
No, Prof. And this is outside Trinity College in Cambridge with his wife, daughter, and granddaughter, and my 4 children on one of the visits. My parents live 10 miles from Cambridge, so it was easy to go over and visit. And so here are the young kids and the professor, and that he was a human being too, and there was another side to him. And he wasn't just a brilliant scientist, he actually truly loved and cared for his family. And so if he can do it, we can. But I asked my daughter, Emily, who now has a spa in Chicago, what she remembered of that picture. And she said, oh yes, we had chocolate cake And we played in the backyard. And the backyard, of course, is Trinity Great Court or something
Right.
like that. So they just thought he was a nice grandfathery type guy. That's another view of Huxley and of the balanced, brilliant life. So,
Brenda, thank you very much for spending the time with me and with our viewership to talk a little bit about your career. It has been a pleasure for me to know you. It's been a pleasure to learn about your life. And on behalf of myself, on behalf of the Society, I wanted to say thank you for taking the time to sit here for the Living History program.
my pleasure.
Thank you.